Glycolysis. A series of reactions that splits glucose molecule 6C into two molecules of pyruvate 3C occurs in cytosol, cytoplasm, can occur with or without the presence of O2. Involved 10 steps that can be divided into two phases. Energy investment phase. Energy payoff phase. ATP is generated in glycolysis by substrate-level phosphorylation, in which an enzyme transfers a phosphate group from a substrate to ADP, forming ATP. Phosphorylation, addition of a phosphate group to an organic molecule. Phase 1. Energy investment phase. Convert a molecule of glucose 6-carbon into two molecules of glyceraldehyde, 3-phosphate/phosphoglyceraldehyde. Involve step 1 to step 5. Phase 1. Energy investment phase. Step 1. Phosphorylation. Glucose is phosphorylated by ATP, forming glucose, 6-phosphate catalyzed by hexokinase. ATP is used to phosphorylate glucose and make it more reactive. Step 2. Isomerization. Glucose 6-phosphate is rearranged, isomerized to its isomer. Fructose 6-phosphate step 3. Phosphorylation. Fructose 6-phosphate is phosphorylated by ATP, forming fructose 1, 6-bisphosphate, catalyzed by phosphofructokinase. Step 5. Isomerization. DHAP is isomerized to glyceraldehyde 3-phosphate. As a result, two molecules of G3P are produced during energy investment phase. Glyceraldehyde 3-phosphate is used in the next step. Phase 2. Energy payoff phase. Conversion of two molecules of glyceraldehyde, 3-phosphate into two molecules of pyruvate, 3C. Involve step 6 to step 10. Step 6. Oxidation and phosphorylation. Glyceraldehyde 3-phosphate, G3P, is oxidized when it transfers its electrons to NAD plus NAD, plus is reduced forming NADH plus H plus. Using energy from this exergonic redox reaction, an inorganic phosphate from cytosol is used to phosphorylate G3P, forming 1,3-bisphosphoglycerate. Step 7. Substrate-level phosphorylation. A phosphate group is transferred from 1,3-bisphosphoglycerate to ADP, forming ATP, via substrate-level phosphorylation 1,3-bisphosphoglycerate, forming 3-phosphoglycerate. For each glucose that began glycolysis, step 7 produces 2 ATP. Step 8. Isomerization. 3-phosphoglycerate rearranged isomerized to 2-phosphoglycerate by relocation of the phosphate group from 3rd carbon to 2nd carbon, step 9. Dehydration. 2-phosphoglycerate is converted to phosphoenolpyruvate by removal of a water molecule. Step 10. Substrate-level phosphorylation. A phosphate group is transferred from phosphoenolpyruvate to ADP, forming ATP, via substrate-level phosphorylation. Phosphoenolpyruvate, forming pyruvate. Since this step occurs twice for each glucose molecule, 2 ATP and 2 molecules of pyruvate are produced. Yields of glycolysis from 1 molecule of glucose: 2 ATP net, 4 ATP produced during energy payoff, 2 ATP used during energy investment. So net ATP produced equals 2 ATP to NADH plus 2 H plus 2 molecules of pyruvate